Okay. I want to thank Carol and all of the people who put this together. It's been a really terrific conference. As always, it's so great to see the researchers and the families come together. It's really great to see Xtraordinaire and ACD both represented here, and I've loved seeing the kids. It's just the best part for me as a neurologist and a mother. So I'm just going to give you a little bit of background on our program at Ultragenyx. We are developing a potential treatment called UX068, and it is currently in the non-clinical part. And so I'm going to talk a little bit about how we work to learn from families, and how the natural history study, Vigilant, which was just completed and presented at the ACD meeting a few months ago, how that all sorts of feeds into the drug development process. Okay, so we are Ultragenyx. We are a rare disease pharmaceutical company started by Emil Kakis in 2010. Emil has made it his life work to bring treatments for patients with rare diseases into the clinic and to attempt to get approval. I'm going to talk a little bit about how challenging that is. We are based primarily in Nevada, but we have offices in a lot of different parts of the world, including Switzerland and France and all over the place. So we are a small but growing company, and we believe very strongly in meaningful relationships with the patient community in every aspect of our work. So disease development is a complex process, as you all know, and it is particularly complex in rare diseases. And there are many reasons for this, including a lack of natural history data. So in other words, we don't necessarily know how kids progress from year to year in terms of their intellectual development or their language development in a way that's meaningful to apply to a clinical trial. So if we are able to bring a drug to clinic and we want to measure its effect on patients, we need to know what the trajectory is for those patients without any treatment intervention, so that we can tell if our drug is making a difference. And this is a critical part of research. And so I know everyone's impatient to get a drug in the clinic, but there are just so many steps we have to go through to get there. And learning how the patients function and develop is part of that process. There's also it's a rare disease by definition, so it's hard to find enough patients. If you're studying an antihypertensive, you can easily go out and you can probably find people, on this block or in the cafe across the street, like 100 patients with hypertension or something like that. But with rare disease, it doesn't work that way. We have to look a lot harder for those patients. And there is extreme heterogeneity. I don't know what that meant. I don't know if I just did something wrong, but anyway. So there's heterogeneity, and what that means is that, as you've seen with the kids running around here, no two are the same. And this is something I learned in Parkinson's disease from one of my mentors, because that was my clinical specialty, is that he said to me, when you've seen one Parkinson's patient, you've seen one Parkinson's patient. And it's clearly the same for CTD. All of these kids are different and unique, not only in terms of their personality, but in terms of the effects of this disorder and obviously the other creatine disorders as well. So this means there are multiple phenotypes. So just because you have one particular genetic mutation does not mean that your manifestations are going to be the same. We heard yesterday that not all patients have epilepsy. Some have focal epilepsy. Some have generalized epilepsy. The level of cognitive development is different for all these patients. So what that means to us as drug developers is that how are we going to find assessment tools that we can use in a clinical trial to assess these patients when every single one of them is different? So that's one of the reasons we have to do so much background work to understand these disorders. And there's a lack of established standards, diagnostic tools, and treatment endpoints. So this means when we are going to take a treatment into the clinic, we have to go talk to the FDA and the other agencies and get them to agree that what we're measuring is significant for the patient population. And I was very naive when I came into the world of pharma, and I'd done a lot of clinical trials. I was a PI or a sub-I on dozens and dozens of clinical trials for Parkinson's and other movement disorders, but I really didn't realize how important that interaction is and how challenging it can be. So we have to have tools we know will reflect what families and kids are dealing with, so that if we can make them better, we can measure that in a way that is not only significant for the patients, but also significant for the agencies if we are trying to get a drug approved, and it's very challenging. So all of these things are challenging, and they're even more challenging in rare disease. All right, so there are 4 basic stages of disease drug development, and the enlarging arrow at the bottom represents the degree of patient involvement and family involvement as the program progresses. So in the beginning, in the lab looking for different formulas that might work, we don't necessarily get a lot of input. We get some input at that point, but we don't get a lot of input. But as the process goes on and we have questions like, if we make an oral formulation, what's the best way to deliver that to your kids? That's when we come and talk to the families and say, as the oral medicine survey we just did with ACD, can they swallow pills? Can they swallow capsules? Can they swallow medicine mixed in with food? What types of food? Do they have certain likes or dislikes about foods that would, like if we say, okay, you can only mix this drug in chocolate pudding, and you all say, my kid hates chocolate pudding, then we're in trouble, right? So that's why it's so important to have that interaction between the families and us as we are trying to develop these drugs. So at each stage, we ask different questions, we get different information from families, and there are people in this room that have really helped us out with that, and we really appreciate that. And we will be coming back for more information. You can guarantee it. So where are we with this preclinical research? So this is the part that's frustrating for all of us, is that there is so much that has to be done in terms of determining whether the drug is safe, whether it's effective, if it gets creatine into the brain, how much creatine do we need? We just heard yesterday, we don't know how much creatine we need to get in the brain to improve function. But we also saw some very encouraging results with the GAMP patients that if you do get creatine in the brain, especially early, it can make a huge difference. So these are all the things we have to consider when we are in the clinic, in the preclinical stage, and that's where we are now. And that stage takes a long time. So you can see that we have to develop the molecule. We have to choose. I think we had started out with over 250 potential prodrugs that we looked at to choose the candidate molecule for this program. We have to explore opportunities for optimal drug delivery, whether it's going to be IV or oral, and there are a lot of things that go into that in terms of safety, ease of use, patient tolerance, et cetera. And we have to do a lot of toxicity studies that are mandatory in any drug development process, especially with the unique and brand new small molecule. So we are slogging through all that. We know you'd like it to go faster. We would like to go faster, but it's just kind of the way things are. Now, this is the sobering slide. So this slide shows the probability of success for various indications. And you can see that the purple box is the neurologic indications. So the probability of success is quite low for neurologic indications, and that's challenging. And as you all know, not every drug that gets studied, not every drug that's developed in the lab, not every drug that goes into the preclinical or the non-clinical work, and not every drug that goes into the clinic is going to be successful. So we are doing the work we have to do to make sure that there's the biggest likelihood of success if and when we get to that point. But it is a challenge. Okay, other key issues and challenges in developing a treatment for CTD include, lighting is not really great on my slide, I'm sorry. There are inherent challenges in studying neurologic developmental conditions. As I mentioned, the heterogeneity of symptoms, that often, as we heard throughout this conference, a delay in diagnosis, although it's wonderful to see that that's getting better, that the time from birth to diagnosis is getting shorter. The lack of natural history data, which the Vigilant was an important part of trying to solve that. Lack of validated measures. So one of the problems we have is that we're using some measures in some of our studies, like the Bayley Developmental Scale. Well, this was developed for neurotypical kids, ages up to 48 months. So you can see that in some ways we're trying to put a square peg in a round hole by using these tools to measure somebody with CTD when it was not developed for that purpose. And I think a lot of the work that Dr. Carey is doing is to try to figure that out and figure out which of these assessment tools will work in these kids. And that's a lot of work that was done in the Vigilance study as well. So we're getting better with our square pegs and round holes, but it's still a challenge. General implications for subject recruitment and retention. We know that you all have busy lives, that taking care of these kids can be challenging. You may even be dealing with more than one child that has CTD, or a child with another developmental disorder. I've met families where they, through adoption or whatever, they've got 3 kids with neurodevelopmental disorders of different etiologies, and that is challenging. So it's hard to be in a clinical trial. Being in a clinical trial is really hard work. You have to show up for these appointments. It's inconvenient. You may have to watch somebody, help hold your child down to get blood drawn. It's really a challenge. So it's something we're very aware of. We're trying to make it as user-friendly as possible, balanced with the amount of information we have to get. And then as we heard yesterday, CTD affects males and females differently. So we're going to have that to take, if we include females in the same trial as males, we'll have to take that into consideration when choosing assessment tools, because otherwise it'll be difficult to show an effect across the population. And we also know that CTD can be missed in early development. It's a relatively new condition. Obviously, everybody in this room is intimately familiar with it, but we all know that there are kids out there or in countries where they can't get access to testing and good pediatric neurologists and geneticists where they are probably not being diagnosed. So that's another challenge. So we have a patient find team dedicated to finding patients for our studies, but it's still very challenging to do this. We also have to look at dosing ranges, the impact of symptoms on ability to participate in the study, and the prevention of symptom treatment if we can, the preference for symptom treatment, I'm sorry. And that's why we did the oral study, because we want to know if we develop a treatment, if your child is going to be able to tolerate that treatment. So we are now, as I said, conducting the preclinical work on UX068, which is an investigational treatment, has not been in the clinic yet in patients. No promise that it will be, but we're really hoping that it will. A key focus on conducting appropriate studies to enable a future investigational new drug submission. We do not have a date for that. I know that's a question in everybody's mind. We don't have one. We will let you know as soon as we're able to release that information. And this will be a first-in-human clinical trial. We are collecting insights in partnerships with advocacy groups, engaging with CTD families like you in multiple countries to understand their experience living with this disorder, and generating data to inform our patient-focused decision-making and plans at every stage. We have completed the Vigilance study. As most of you know, we took this study over from Lumos. I think it was 2017, 2018, a natural history study in 50 males with CTD. If I had been able to design that study, I would have said, and females, but it didn't. I got it when it was already a fait accompli, and I know Simona Bianconi, who some of you know, really wanted to have females involved as well, but that it didn't happen. So we inherited the study. We went forward with the study. We got some really great data from it, but we don't have data from that study on females. So it's one of the reasons I was so excited to come to this conference and hear more about CTD in females. So some of the key research questions driving our preclinical activities is, what is the best way to deliver enough prodrug into the brain to achieve optimal levels of cerebral creatine? How does the drug get absorbed, distributed, metabolized, and or excreted? So no toxic residues build up or anything. What oral formulation is the safest for patients and the best tolerated? What are the potential dosing ranges. So how big of a dose can you get, and how often do you have to give it to get optimal creatine into the brain? So there's research underway to address these questions, currently testing various dosing parameters in single or multiple daily doses, giving the drugs to, I'm sorry, there's feedback, right? This is the lighting is so giving the drug in a fasted and fed state to find out ultimately if we're able to go into the clinic, will this be something that we give with food or on an empty stomach? And then the safety and tolerability of the prodrug formulation for dosing, oral dosing is tested on dedicated toxicology studies that will be purpose designed. So we will design studies specifically to get the information we need from those non-clinical studies, and also looking at what happens when you dose this chronically over a long period of time. So we also want to know how the drug is absorbed, distributed, metabolized, and excreted, and examined to some extent in each animal study that we conduct. So we are also conducting dedicated mass balance studies, which allow us to measure the prodrug and any metabolites in the blood, exhaled in air, tissues, urine, etc., so that we can see what proportion of the dose is distributed in different parts of the animal, and ultimately, we hope, the human. Okay, so as I mentioned, CTD community engagement is very important. The vigilance study was a great example of that, where these dedicated families brought their children in for those visits. Some of them went to the NIH to get additional scans, blood tests, skin biopsies, etc. A lot of work on the part of the family, a lot of dedication, for which we're all very grateful. And then we are in the process of, we've completed a clinical study report, and we're now in the process of writing up the study manuscript, and that will be published and available for anyone doing research in CTD, whether it's Ultragenyx or someone else. And I think it'll be very helpful. And I, of course, many times read Jiddick's paper on 101 male patients with Parkinson's. And it's, I think, what the data that we've gleaned has been sort of a nice correlate to that. That was a single look, and this study looks at patients over time. So all this work is foundational, and then each study builds on it, on the ones that were done before. And then burden of disease study we've done with advisory boards, had advisory discussions. We have a CTD Caregiver Leadership Council, and some of the members are here with us today, which has been really great, and one-on-one interviews with caregivers, and as I mentioned, the oral medication survey that was conducted by the ACD, which we are kind of now ingesting internally to figure out what we can learn from that, and what's the best way to analyze that data. So just as some additional background on natural history studies, it gives us an idea of disease progression. Do things get worse over time? Do they stay flat? Do they get a little bit better over time? We need demographic, genetic, or other variables that impact outcomes. For example, if patients live in an area where they can't get to a medical center for, say, a clinic visit or a clinical trial visit, we need to know that. How can we address that? How can we try to make it easier for families to participate? We don't have as many sources of information as more common diseases, as you all know, and so we have to have a more planful and sometimes more creative approach. We also get information on disease progression and skill development over time. We help identify appropriate patient populations that might participate in a future clinical research trial and suggest which assessments might be appropriate for a population. This can sometimes be used as external controls in a clinical trial, although I will tell you that's very challenging. So the agency generally wants to see, once you get to like a pivotal trial, they are generally most likely going to want to see a placebo-controlled study. But sometimes you can take that natural history data and in some cases use that as control data. Very hard to do. And then also some of the blood tests and imaging and things like that from the study might enable development of a biomarker that can be used in the clinical trial to assess drug effect. So what have we learned from the vigilance study? we learned that patients with CTD showed significant and persistent impairment across all constructs measured, including cognition, behavior, communication, receptive and expressive language, motor function, daily living skills, and interpersonal skills. And the assessments demonstrate consistency across visits. So the implications for natural history are that 2 years provided a subset of assessments that were very reliable in this population. And the implications for drug development is that we have several assessment tools that should help researchers interpret whether a treatment is or is not effective. So that was another very important part of the Vigilance study, was to say, start with a big list of assessments and tests and figure out which ones work in this population and which ones don't. And we were able to eliminate, with the first round of data review we did, we were able to eliminate certain assessments that just don't seem to work in this population. And then we also are working a lot with gross scale values, and this is a way of analyzing data so that instead of saying, how does this patient compare to a neurotypic child, or how does this patient compare to all the patients in the study, we say, how does this patient compare to themselves? So how can we measure their change over time in that particular patient? And this is a very helpful thing to do with such a heterogeneous disease where the patients are also different. Okay, things we tried that didn't work well in the study were asking children to wear things like trackers. So I'm seeing some knowing looks. Okay, anybody surprised by that? So I really like the idea of wearables. I think it's a great idea in principle, but in practice, I know it was challenging, and most of the kids did not like it. In another study I did, we asked the kids to wear a watch in GLUT1, and they were complaining that the color was boring and it didn't have any games on it. So a lot of kids just don't like the wearables, unfortunately. So following precise directions, verbal or nonverbal, it was difficult to get consistent performance with limited receptive language capabilities. And I'm probably not telling you anything that isn't obvious to a lot of the parents in this room, but sometimes the kids don't follow verbal instructions, and especially motor tests. Like, the FDA is big on the 6-minute walk test, and you cannot tell these kids, okay, we want you to just walk back and forth between these cones for 6 minutes, because it's unlikely that they're going to do it. So there were a lot of things we were able to eliminate in the study. And then, as I mentioned, the rating scales are designed for non-intellectually disabled kids. And so, like, for example, the agency likes the ADHD rating scale and the parent stress index, but neither of those was designed for kids with developmental disorders. So they just don't fit the purpose. And then Vigilance provided the opportunity to gain more information about the progression of CTD in children and adults. We did find that there was some improvement in learning, like very small improvements over time in some groups of kids. But there'll be more about that in the manuscript when that comes out. We're still analyzing that with the team at the NIH to get more granular information on that. But we did definitely gain more information about that, and we hope to further characterize the disease and study population, and these learnings will provide help us make decisions for future interventional studies, as well as other workers in the field. So it was a really great opportunity to be able to finish that study. So the next steps, as I mentioned, there's a manuscript in preparation. There is a clinical study report. We are planning to release a plain language summary for families. At this point, you've probably read enough about this that you don't need the plain language summary, but there will be one. And thank you very much. And again, thanks for all the participants in the study. So.